Hello, welcome to MiceTech Talk with the Technical Information Scientists at the Jackson Laboratory. My name is Dolores, and my job as Technical Information Scientist is to serve the research community by answering all mouse-related questions.
And my name's Caitlin. MiceTech Talk is a way for us to connect with the research community and answer some of those mouse-related questions live. And Dolores and I are joined today by our newest colleague in TIS, Paige. So welcome to TIS, and we're happy to have you on MiceTech Talk this week.
Thanks for having me. I'm really excited. So hopefully you're all ready with your questions because it's time for MiceTech Talk. So today, let's talk exploring neurobiology solutions.
In this episode, we're going to be discussing exciting neurobiology research that's been conducted here at JAX. And we had planned on presenting this information at the 2020 Society of Neuroscience meeting, which you all know was canceled. So we are actually very delighted to share this information with you virtually. And we're going to share some of the highlights of JAX's preclinical research studies using mouse models for ALS, Alzheimer's, and Friedrichs ataxia.
For you watching live, you can definitely use the comments section of the video to tell us where you're viewing from, if you're bummed out about missing SFN this year like I am, and any other neurobiology related questions that you might have. And we'll try to answer those throughout this session.
That's right. I see we have somebody watching from Kalamazoo. That's great. Welcome. So Friedrichs ataxia was the first talk given by Kat Latz. Caitlin, what were your thoughts on that one?
Sure. So first of all, for those of you who don't know, Friedrichs ataxia is an autosomal recessive disorder that's characterized by an expanded trinucleotide repeat in the first intron of the frataxin gene. So it's a GAA repeat. And this repeat leads to a decrease in the amount of frataxin protein that is expressed and ends up leading to an increase in iron in the mitochondria while causing a decrease in iron in other areas. So at JAX, I was really excited to hear about the two models that we have to study Friedrichs ataxia. The first one is a Phlox model. And these mice were generated using CRISPR-Cas9. So they're a Cre-conditional frataxin allele model. And this model seemed really useful when studying the different phenotypes of the disease because we can use methods such as gene therapy to modulate the phenotype. So this model, the phenotype was really heavily present. However, in the other model, where we knock out the mouse frataxin gene and then knock in a human transgene, so the mice are homozygous for the frataxin knockout and then hemizygous for the frataxin human transgene. They display sort of this age-dependent, tissue-specific GAA repeat. But the really interesting thing is that these mice don't display any sort of phenotype. So this model is really good for studying therapeutics that might increase the amount of frataxin protein.
Great. So one of the questions that was asked during that talk was, why do the mice tolerate low levels of frataxin protein so well? Can you give us an insight on that question?
Yeah, absolutely. So this is an area of ongoing investigation for sure, but the best answer that I can give is that these mice must have some sort of compensatory mechanism for low levels of frataxin. But the exciting thing is that if we can figure out what those compensatory mechanisms might be, we might be able to use those mechanisms to better treat patients who have Friedrichs ataxia. So before we move on to talk about ALS, I just wanted to remind everyone to submit your questions so we can get them answered. So I don't want you to wait till the end of our show because we might not be able to get them all answered. So Paige, I know you were really interested in the ALS phenotyping strategies talk that was given. What did you have to say about that?
Yeah, so like Friedrichs ataxia, amyotrophic lateral sclerosis, or ALS, is also a devastating neuromuscular disease. And so here at JAX, we have a number of ALS mouse models and phenotyping assays that we use to kind of characterize new models, as well as testing potential therapeutics. So one of the challenges in this field is that many of the therapeutic candidates that have been successful in the mouse models really haven't conferred significant benefit to our human patients. And so there's really this interest and need to keep developing new mouse models as well as more sophisticated ways to study them. So if you've ever worked with ALS or other neuromuscular disease models like I have, then you know that there are pretty significant limitations to the traditional assays in detecting the disease manifestation, as well as modification in response to treatment. So what was shared the other day is that the group here really kind of started to think about how to design more appropriate in vivo assays. And one of the things when kind of thinking about how to put these new assays in place is really, are these going to be clinically relevant? And so the new assay that Dr. Bogdanek shared in the neurobio session is adapted from the electrophysiological tests that clinicians use for diagnosing and tracking ALS progression right in human patients. And so this compound muscle action potential, or CMAP, and repetitive nerve stimulation, or RNS, are set up and used in our live mice very similarly to the way that ALS patients are tested in the clinic. And so really what this test does is kind of look at the function of the motor unit. So what I thought was like really exciting about this is the sensitivity of this assay. The onset or the changes in ALS models can be detected even before axon loss is prevalent. And so that's pretty early, the axon loss in the histopathology. But many of the other in vivo assays that are traditionally used are much later. Those can't detect the onset until much after all of the neurodegeneration has occurred. And so what's even more exciting is some data that Laurent shared showing the preclinical trials of a therapeutic compound in a well-known ALS model. And so with the more traditional assays like grip strength or rotarod, the therapeutic benefit of that compound was detected in the later time point or not at all. But the CMAP was actually sensitive enough to detect changes in response to this compound. And that was sensitive enough to even detect it at the earliest time point. So what I think that this really shows is that using these electrophysiological assays like CMAP and RNS in your ALS pipeline and testing new therapeutics or new mouse models for ALS really can be an asset for detecting candidate therapeutics that you otherwise might have overlooked, especially because if you lacked sensitivity in your overall pipeline. And so while it's really exciting to see these new assays that are being put in place here at JAX, Dolores, I know that you were really interested in the new Alzheimer's mouse models and the exciting collaborative preclinical pipelines that Model AD shared.
Absolutely, absolutely. So Gareth Howell gave a fantastic presentation summarizing what Model AD consortium has done so far, generating not only new mouse models for late onset AD. He also described their phenotyping pipeline to test compounds to treat Alzheimer's disease in mouse models. And also he summarized the progress made looking into genetic modifiers as well as environmental factors to modify and aggravate the phenotypes seen in a number of different mouse models of Alzheimer's. So they have standardized a pretty impressive phenotypic pipeline, first of all, using the familial model 5XFAD at different ages. And the nice thing, similar to what you were saying, Paige, is that they are also using clinically relevant readouts, of course, for Alzheimer's disease. He highlighted results using in vivo brain imaging, using an amyloid PET tracer, transcriptional profiling to see how a specific sets of genes, their response aligns to transcriptomes seen in patients with Alzheimer's to see if there was a match there or not. He also showed some results looking at neuropathology to look at microglia activation and the assessment of a number of blood-based biomarkers such as amyloid beta 140, amyloid beta 142. These studies are run by the Preclinical Testing Core of Model AD. This core is co-led by Paul Territo from Indiana University and Stacy Rizzo, who used to work at JAX and now is at the University of Pittsburgh. They are the ones in charge to set up the pipeline and do all of the studies. Dr. Howell showed very nice results testing the pipeline, specifically use it for to looking at the efficacy of verubecestat, a BACE1 inhibitor in the 5XFAD model, and using those readouts that I mentioned before. And so the one thing that I wanted to mention is that for more details on about the pipeline and the readouts that they use, I invite you to attend a JAX webinar that's happening next week, next Thursday, Improving Translational Relevance for Preclinical Mouse Models of Alzheimer's Disease, November 5th, where I talk about that in a lot more detail. Very exciting for everybody in the space working on potential compounds to treat Alzheimer's disease. Gareth also featured the Stop AD portal, which allows the greater research community to nominate compounds to treat Alzheimer's disease using their amazing pipeline and using the most appropriate mouse model, depending on what compounds they are trying to test, and to see if those compounds are efficacious in reverting a number of Alzheimer's disease phenotypes in these mouse models. So researchers can submit their compounds, which are going to be reviewed by a committee, and the study will be totally funded by the NIH. So this is an amazing opportunity that you can look into. On the other hand, he described the phenotypes of the new late onset models of AD that were generated by this consortium. And he discussed a number of approaches to investigate, as I said at the beginning, not only the genetic components, genetic factors, but also environmental factors that may contribute to Alzheimer's phenotypes. He showed evidence that wild-derived genetic backgrounds can accelerate the neuronal loss and changes in microglia and astrocyte response to amyloid deposition compared to the same mutations on a BLAC6 genetic background. So there's a lot to investigate about that. Also, this group is doing very interesting work putting many of these mouse models on a high-fat diet, high-fat, high-sugar diet for at least 10 months, and seeing interesting phenotypes that are exacerbated, such as increased cholesterol in blood, and they are, ongoing studies to see how that may impact the Alzheimer's phenotype. So finally, just point to the fact that all of the data that has been generated is uploaded in the AD knowledge portal, as well as all of the very descriptive protocols that they're using for all of these tests. So I invite you to go there and check that out. So I'm going to stop here and let me check for some questions from our viewers. We have a lot of people from different parts of the world. Let me see. This is one for Paige. Let me see. Well, let me read it first. Hold on. The PFN1G1 aiding V is one of the new mouse models for ALS that JAX has. Have you done any further studies on this model?
Yeah, it's a great question. So we're always further characterizing new models. I'm not sure if the PFN1 has gotten into any of the pipelines in Kat's group yet, but we can definitely look into that for you. All of the data that Dr. Laurent shared for the CMAP data was actually done on the SOD1 G93A, which is one of the most commonly used ALS models. But we do need better and new models. So it will be exciting to see what comes out of that model and others.
Great, great. So let me see. That's, I think, all of the time we have for questions this week. We have linked a few resources in this video description. Caitlin, can you quickly comment on what those links are?
Sure, absolutely. But before I do that, I saw in the comments, I can't tell what or who posted this, but someone just defended their thesis and they're looking for a job. So congratulations and good luck in the job hunting.
Yeah, there's tons of job opportunities at JAX, that's for sure.
Yeah, absolutely. You can check out our job postings page. We're always looking for new people. So the links. The first one is the Exploring Neurobiology Solutions page, and that link will allow you to register for tomorrow's scientific session. So we had one last week. That's where all of our information came from today. So if you're looking for more information or you want to ask some of the scientists who did the research questions, you can do that. So go to that link to sign up. The session is tomorrow. We also have a link for the Neurobiology Resources by Therapeutic area. So if you're interested in a particular disease area, head to that link. We also have our Neurobiology Research Tools and Online Resources page, which is a great page if you're just looking for some more general information on different neuroscience topics. And then finally, we also added a link for Dolores's webinar next week. So it's improving translational relevance of preclinical mouse models of Alzheimer's disease. That'll be November 5th at 1 p.m.
Great. So our next MiceTech talk is called Let's Talk Your Favorite Database on Tuesday, November 3rd. You can go to LinkedIn or YouTube to watch it. And we look forward to seeing you next week. This is Dolores saying stay healthy, stay safe, and stay excited about your research.